Okay. Hello, everyone. Thank you so much for joining today. Today we have one speaker. His name is Jae Kyoung Kim. He's an associate professor in the Department of Mathematical Sciences in KAIST, and also he's the chief investigator of the Biomedical Mathematics group in IBS. He got his PhD in applied and interdisciplinary mathematics at the University of Michigan, and was a postdoctoral fellow in the Mathematical Bioscience Institute at the Ohio State University. He has combined nonlinear dynamics, the theory of stochastic processes, and scientific computing to solve critical biological and medical problems. With this short introduction, welcome to this week Crunch seminar. Jaya, thank you so much for joining us, and you can start.
Yeah. Hello, everyone. First, thanks so much for giving me a chance to share our work in this exciting seminar series. Today, I'm gonna talk about two things. We recently developed the density pin, and then I will explain what it is, and then after that, I will describe how we use this density pin to analyze some biological data to serve some key mystery in biological system. And this work is done by is done with my talented graduate student Jae Kyoung, who is current Ben Black postdoc fellow in Wisconsin, and senior researcher Hyun Tae-jo, who will begin assistant professor in Korea University soon. So let me begin. So first, let me describe the basic concept for the cellular signaling pathway. So we have a cell, and in cell has a membrane. And if extracellular signal arrive to this membrane, then cell can sensing this signal, and this signal is transduced through this complex pathway. And then when it arrives to some finer places, it will generate some response molecule. So it could be typically it's some gene transcription. So for instance, if you look at this image, if we give a certain drug to the bacteria, at the beginning there's nothing happen, but as time goes, you can see that yellow light is emitted from the bacteria, which indicate that some specific molecule is produced at the bacteria. So in the experiment, by measuring this light intensity, we can measure how many number of the final response molecules are generated throughout the time. At the beginning, it's 0, but as time goes, you can see that the number of the molecule is increasing, which is induced by the extracellular signal. So, and you can see that there are about 20 molecules are generated in this figure. So let me call the first molecule, the generation of the first molecule time is tau1, and then second molecule generation time is tau2, and third is tau3 and tau4. We call this is signaling transduction time, because this is the time for the molecule to be generated. And then if we merge this time, we can get the distribution of the time. This is we call signal transduction time distribution. Okay. So by using this signaling transduction time, we can simplify this complex signaling pathway to the simple delayed burst system. You can see that the initiation of signal, we can think that there is a Poisson burst process with the rate of the lambda b. And then this intermediate pathway we can replace with this time delay distribution. And then final molecule decay is also described by this death process. So we can think this is queuing process. So burst initiation occur with this Poisson process with the lambda b. And then as long after this initiation happened, the time for the molecule generated is following this time delay distribution, and then that process occurs. And then by doing some mathematical analysis, we can show that the mean of this yellow molecule here following this delayed birth death process. Okay. And this delayed distribution doesn't need to be the unimodal distribution, but sometimes it could be bimodal distribution or trimodal distribution, but it can be various shape. But importantly, the shape of this time delay distribution has a very important information of the underlying network structure. If it has a unimodal distribution, then it indicates that the pathway is a single time scale pathway. But if the distribution is bimodal, what that means is there is some group of the molecule which is generated in the short time scale, and another group of the molecule which is generated in the slow time scale. And in this case, we can imagine that there are two pathways. One is a very fast pathway, and another is a slow pathway to make the molecules. So if we know this time delay distribution, shape of the time delay distribution, then we can see the structure, we can infer the structure of the network. So, okay, but there is some challenging. So here are the simulation results. So here is a single time scale pathway, which has a unimodal time distribution. And here is a multi-time scale pathway with a bimodal time delay distribution. And then here is the output of this signaling pathway. So here only YT is observable and GT is not observable. But if you look at the time series data YT, you can see that these two time series are nearly indistinguishable. So from this indistinguishable time series data, we have to infer this non-observer time series data. So this is quite challenging problem. So for this, that means mathematically, from the YT time series data, we have to infer the function g of s, which is inside of this integral. As you know, usually integral smoothing out the information of the GS, so it's not easy to infer the function of GS from the time series data. So typical optimization algorithm doesn't work to infer the function of G of S, so that we decide to use the neural network approach. So, so typical neural network approach using the cost function of minimizing the solution and observed time series data. In that case, you can see that when they're in the places where the data exists, the fitting is pretty good, but outside of the data, the fitting is not good. So that one way to overcome this is tell the governing equation to the neural network by introducing this additional residual term, which is satisfying the given governing equation. And this is called physics-informed neural network, as everyone knows in this seminar series, and developed by the George Kaniadakis. And by just telling this physics, then you can see that even outside of the data point, the fitting is very good. So we decide to use a physics-informed neural network for this problem, but one obstacle was we need to infer the function g of t rather than some specific parameter of the system. So we were not able to use the pin directly. So we have to extend the original pin to estimate infer the function rather than the parameter. So for this, we decide to use the kernel. So specifically, we decide to use the Rayleigh distribution as a kernel. So Rayleigh distribution is one of the probability distribution which has a positive support, because we will infer the time delay distribution, which always a positive value, so that Rayleigh distribution was good candidate. And then Rayleigh distribution has a two parameter. So CJ determine when the non-zero part beginning, and Sj determine the variation of this distribution. And what is well known in the statistics is for given any probability distribution, and if we have enough number of the Rayleigh distribution, we can always make a good approximation for the original distribution. So Rayleigh distribution is can function as a corner. So so that for given any time delay distribution, we can express as the weighted sum of this Rayleigh distribution. So now we want to infer the time delay distribution GT. Now we just need to infer this weight omega j and Cj and Sj. Then our game is over. So the key is now we change the function inference problem to the parameter estimation problem. So for this, we will infer the omega j and Sj. Sj determine the variation of the each Rayleigh distribution, but we will use the fixed Cj. That is, we will make a uniform of this uniform distribution of Cj across the some time domain. So we now just need to estimate the scale parameter Sj and weight parameter omega j rather than function of the whole time delay distribution. So okay, here is let me explain our density pin. So from given time series data y of t, we will use variational autoencoder because our data is has a various noise due to as its experimental data. So we decide to use a variational autoencoder to estimate the omega and s omega parameter and s parameter. So from this omega and s parameter, we can construct the time delay distribution. Okay, so then from this time delay distribution GT, now we have to decide a loss function for the scale parameter Sj and omega j. One simple way is if we know the true distribution GT, we can compare the estimate G tilde T and GT, but unfortunately GT is unknown. But only we observe the Y of T. So that what we have to do is from this estimated G tilde T, we serve this integral differential equation that we can get the solution of this integral differential equation we call Y tilde T. Then rather than lost function data loss is GT and G tilde T, we can make YT and Y tilde T as data loss. So if we make the loss function so that Y tilde T go to the YT, then it implies that G tilde T will go to the true G of T. But one problem is this convolution. In this differential equation, there is a convolution which makes this calculation is very inefficient. So that we need an alternative approach to overcome this. For this, what we will do is we will make an approximate solution for this original differential equation we call Y tilde approximations. So you can see that it's just a weight sum of the integral. So there is no more convolution term. And we will use this Y tilde approximation rather than original Y tilde as data loss term. However, Y tilde, this approximation does not need to satisfy this differential equation so that this data loss does not imply that Y go to the Y tilde, so that it does not imply that G tilde go to the G. So that remaining job is we have to change this inequality to the equal. So how to do this is we will make this Y approximate solution satisfy this physics loss term. By enforcing this physics loss term, what we can say is Y tilde and Y tilde approximation become similar. So by summarizing this, what we can say is Using the physics loss term, y tilde and y tilde approximation go to this difference go to the zero. And using data loss term, y and y tilde approximation go to the difference go to the zero. By merging this, we can make y and y tilde difference go to the zero, which we originally want. So in summary, from the time series of YT, using the variation autoencoder, we will infer the G of tilde. And then using the another neural network, we will make a Y tilde approximation from this estimated time delay distribution of G tilde. And using the data loss term, we will make YT and Y tilde approximation is close to the zero. And using the physics loss term, we'll make G tilde and Y tilde approximation go to the going to the zero, so that we achieve the goal that throughout this density pin, G tilde go to the G. So now let's use this density pin for some simulated test. So as I said, this G of S can be various type of time delay distribution. So here I consider three cases. One is unimodal cases, another is weak bimodal cases, and another is strong bimodal cases. And here is a simulated time series data from this stochastic simulation time series of this from each time delay distribution. And please look at that this observable output time series data of these three cases are nearly indistinguishable. So with the human eye, it's not easy to tell which comes from the unimodal distribution or which comes from the bimodal distribution of the time delay. So now we will only use this time series data, but without no information of this underlying delay distribution. When we apply the density pin, you can see that it can completely recover the original underlying time delay distribution. Okay. So after we found that this density pin works successfully for various simulation studies, now we decide to use it for the real biological system. So the real biological data we will use is response time series to the antibiotics. So as you know, we use the antibiotics to kill the bacteria inside of our body. So if we give the antibiotic to the bacteria, then what happens is this antibiotic reaches to the cell membrane of the bacteria, then it triggers a certain signal, and then and this signaling triggers the various response molecules. So to kill the bacteria. And you can see that here are about 16 types of the gene which are expressed by the antibiotics. So all these 16 are different type of the gene. So and you can see that there are many trajectory, because here what we measured is after giving a antibiotic to the single plate, and there are about 50 bacteria in this single plate which has the same genome. And we measure each bacteria's responses. So here, single trajectory means a response of each bacteria. And the important thing is, if you look at this left top, although we give a same antibiotics and same amount in the same environment and same E. coli, you can see that their response are very heterogeneous. And actually, this is a big issue for the antibiotics application, because if you look at the type, this bacteria highly respond to the antibiotic. That means it will be killed. But this bottom one, you can see that this molecules are nearly not expressed in this bacteria, so this bacteria will be survived like this figure. And for this bacteria, what happens is after a couple days, this bacteria is grow up so that antibiotics is failed. So knowing that such heterogeneous response across the bacteria to bacteria has been a big problem in this antibiotics successful application. So we wanted to know what is the key for this heterogeneous responses among the bacteria to the same antibiotic. I grouped the gene to the two color. So pink, red color group, you can see that their response heterogeneity is quite high. But the blue one is, you can see that quite homogeneous. And we applied the density pin to each time series data to estimate the underlying time delay distribution. And we get a very surprising result. And the right side is inferred time delay distribution from the observed time series data by using the density pin. Interestingly, this high heterogeneity group has a unimodal time delay distribution, but the low heterogeneity group has a bimodal or trimodal time delay distribution. So what this indicates is, at the beginning, I told you this time delay distribution can be used to infer the underlying network structure. So this unimodal time delay distribution means there is a single pathway, and bottom bimodal or trimodal distribution indicate that there are multiple pathways. So the summary of this work was, if there is signaling pathway with the unimodal time scale, then we can expect that the response to the signal will be very heterogeneous across the population. On the other hand, if there is a combination of slow and fast pathway, then the heterogeneity will be small, so that we will get the homogeneous response to the drug. So this gives a very important message to the drug development. When we develop the drug, we need to identify the target of the drug. Then what will be the good candidate for the drug target is, we should target which is regulated by this multi-time scale signaling pathway to get the homogeneous responses. So for instance, if we want to develop the anti-cancer, which can kill all the tumors, we need to target some gene which is regulated by this multi-time scale pathway. So in summary, so from this finer response time molecules, by using the density pin, now we can infer the time delay distribution underlying the signaling pathway. And from this time delay distribution, we will able to identify the underlying structure of this signaling pathway. And from this, we can tell in which case a cell will show the homogeneous response to the extracellular signal or heterogeneous responses. So this is the first part of my talk regarding the pin. But at the remaining about 10 or 15 minutes of time, I will talk about a little bit different story, which is also our recent work. It's about the causality inference. So inferring the causality from the time series data has been a very important problem in various areas, not only biology, but up to economy or ecology and sociology. So basically, we want to know, just from time series data X and Y, we want to know whether X causes the Y or X regulate the Y. In terms of ecology, what that means is, here is the time series data of the number of the paramecium and dendridium. And just measuring this time series data, we wonder we can infer their prey-predator relationship. So there is a positive regulation from the prey to the predator because prey is food for the predator. And because predator kill the prey, there is negative regulation. So from this time series data, can we infer this regulatory network is the topic I would like to discuss at the remaining of time. So one approach for this is write down some mechanistic model, and then let's fit this model to this time series data. If this delta parameter is positive, which indicate their interaction term, we can say that there is a positive interaction. And if beta is negative, then we can say that, oh, there is a negative interaction. So this is what we call a model-based causation method. But one limitation of this approach is to infer the causation, we have to write down the equation first. But usually, when we need a causation inferences, this is usually the case. We have little information about the system. In that case, writing down the equation for the system is nearly impossible. Furthermore, even though we know the equation, if the system is complex, that means we have to fit the many parameters, so it causing the high computation cost. So to overcome this issue, a couple of years ago, Cindy was developed. What is this Cindy? Is rather than write down the differential equation, they list up the alphabet for the equation like this, and then they served some linear algebra problem. So they determine the coefficient of this alphabet equation so that they can infer the mechanistic model without writing the equation. But Cindy has a still problem. One is, anyway, there is a limit of the alphabet. So what if the system cannot be described by this alphabet, then it can cause the false prediction. And number two is, although they use this sparse optimization technique, but still identifiability issue was frequently happen if you try to use the Cindy. So that we have decided to use a completely different approach. So we call it a general model-based inference. So what is the general model inference is? We will not write down any equation. So we will just write down most general form of the differential equation. In the 2D cases, it will be f of x and y, and g of x and y. And f and g could be any smooth function. So although we don't know what it is. And then from the tool we will develop, which I will describe, we will determine the sign of this each function. So df over dx and dg over dy. From this sign, we can tell there is either a positive or negative regulation without knowing what is a function f and g. So let me explain what is this general model-based inference is. So, and this work is done with my previous two talented undergraduate students, Sungmin Ha and Seo Park, who is a graduate student now in MIT and Wisconsin from last fall. So, so here, let's begin with the very simple cases. So let's say X regulate the Y. So that means the derivative Y is, for instance, X cubed, but the right side could be any positive, any monotonic increasing function of the X. Then here are some time series data. So what is the meaning of X positively regulate the Y? That means the derivative Y equal to the sum monotonic increasing function of X, right? So that means we can say that whenever X increases, Y dot should always increases. That's the essence of the positive relation of X for the Y dot. So that means for at this time series data, let me pick up any two time points which satisfy this inequality. So let's pick up the time point t and t star, and xt is greater than xt star. So x difference is greater than 0. Then due to this monotonic relationship, y dot t minus y dot t star should be positive as well. This is always true for any choice of t and t star satisfying this inequality. So that means for such a pair of t and t star, if we multiply x difference and y dot difference, their multiplication should be always positive, okay? And we call this is regulation detection function. And if we draw this regulation detection function at the domain where xt minus xt star is positive, then you can see that this regulation detection function is always blue. That means they are always positive. Okay. And then we will calculate the regulation detection score, which is integral of this regulation detection function, which is normalized by their absolute integral function. So this indicates the fraction of the positive area of this function. So in this case, it should be the 1. Although this is a simple idea, but very powerful, because to draw this regulation detection function, we just need time series data. We don't need what specific equation is. From the time series data, we draw this regulation detection function. And if this regulation detection function is always positive, then we can say that, there is positive regulation from the x to the y. Okay, so one more time. From just the time series data, which we can observe, we can make this, we can draw this regulation detection function. Just by checking the positivity of this regulation detection function, we can tell there is a positive regulation from x to the y or not, okay? We can do the similar thing for the negative regulation. In the negative regulation case, the above statement is slightly changed. Whenever x decreases, y dot should increases. So if we change this to the above inequality, you can see that if x difference, xt minus xt star is negative, then y dot t minus y dot t star should be positive. So that means minus xt times y dot t should be always positive. And let's see, really, this it is for the regulation detection function. You can see that if we draw this regulation detection function at the domain where x difference is minus, you can see that they're again always positive. Okay, so that their regulation detection score becomes again 1. Okay. So this idea can be extended to any dimensions. So far, I only consider the 1-dimension input, but now let's consider 2-dimension input. So in this case, the statement is changed like this. Whenever both x1 and x2 increases, y dot should increases. So that means x1 difference and x2 difference are positive, then y dot the difference should be positive. So if that means our regulation detection function is x1d times x2d times y dot d differences. You can see that this multiplication is always positive. Okay. What if we choose the different regulation detection function? For instance, to detect the plus minus regulation, we have to use this x1d and minus x2d and y dot d regulation detection function at this 2 domain. You can see that in this case, not positive. There is a mixture of positive and negative regulation. In this case, we can say that, there is no plus minus regulation. So by just drawing this kind of regulation detection map and just looking at when the always blue happen, we can decide the type of the regulation. And so for the plus minus regulation, you can see that plus plus regulation detection function is a mixture of the blue and red, but plus minus regulation, it becomes always blue. Okay, but sometimes you can see that two regulation function become both are blue. In this case, plus plus is yes, and plus minus is also yes. When this happens, you can see that the second regulation plus and minus coexist in this case. That means second regulation doesn't matter, so that actually the actual regulation is 1D. So by looking at these complete cases, we can remove the one causation from this 2D regulation. So that's all. So now we are ready to apply this framework. We name this as general ODE-based inference, or GOBI. So from this, let's begin with a very simple example. Here is some three-node network where two positive regulation and one negative regulation. And let's simulate the time series from this ordinary differential equation with the various initial condition. And let's begin with the 1D regulation detections. For the 1D regulation, there are 9 types of the regulation is possible: A to the B, and A to the B negative regulation, A to the C positive regulation, blah, blah, blah. So that we have, there are 8 different types of positive regulation. And then for each type of regulation, we will draw regulation detection score for each time series data. And then we see when always positive. And you can see that A to the B negative regulation case is always positive, but other regulation type has mixture of the plus and minus or red and blue. So we can say that they are not true. So from this calculation, we can infer that there is one negative regulation from A to the B. And then we can do the similar thing. We will check all possible two-dimensional regulation. You can see that there are 3 different types of the regulation. A and B both are positively regulated to the C, and A has a negative regulation for the B, and C has a positive regulation. So there are 3 inferred 2D regulation, but we should be careful because first 2 cases, you can see that C to the B has conflicting result. That means this is not correct one, so that only A to the B negative regulation 1D is the case. So this is not. So that there is only single 2D regulation inferred. So by merging original 1D regulation and 2D regulation, now we can infer the original network structure. Okay. So now let's apply this to the real experimental data. So first, let's apply the GOBI to this paramecium and dendridium data. Then we can infer the 2 1D regulation, positive and 1 negative. By mixing it, we can get this negative feedback loop structure between prey and predator. Then what about the molecule? So this is a molecule level change of the 2 type of the molecule in the bacteria. And if we apply the GOBI, there is a 2 1D regulation and 2 2D regulation. If we merge this, we can again infer the negative feedback structure with the self-negative regulation. But why there is self-negative regulation is typically the molecular decay level is proportional to the itself, so that that is came as a negative regulation, as you can see here. So now let's make problem is a little bit more challenging. So here I got the 4 time series data by mixing this paramecium, dendridium, and one of these 2 molecule data. So there are 2 come from this bacteria data, 2 come from the molecule data, so that we have 4 time series data. And then let's plug in this 4 time series data to the conventional causation detection method. So the most famous one is granular causality method. And if we plug in this 4 time series data to the granular causality, you can see that there are a lot of the force prediction. It say that this bacteria causing the molecule and molecule causing the bacteria, there is many wrong prediction. Actually, that's the majority. That's the problem of the previous existing causation detection method based on probability or statistics or information theory. It works easily, but their accuracy is very, very bad. That's the problem of the previous causation detection method. And another famous method is convergence cross-mapping, which is developed by the George Sciarra a couple of years ago. And again, you can see that they also make the false positive prediction between bacteria and molecule. And most recent method is partial cross-mapping method, which is developed by a Chinese group a couple of years ago. You can see that only this method can prevent this false positive prediction. And you can see that Gobi also successfully inferred their two independent regulatory network, but Gobi provided further information over the PCM. That is, previous causation detection method only can tell that there is some causation or not, but Gobi, not only that, it can tell there is a positive regulation or negative regulation. So now let's apply this to the other type of data set. So we apply this to the air pollutant level change for 3 years and cardiovascular disease patient hospitalization number in Hong Kong. So it is well known that NO2 level and respirable suspended particulate number are major risk factor for the cardiovascular disease. And we wonder, just from time series data of this, we wonder we can infer this network structure. And we apply the granular causality, CCM, PCM, and Gobi. You can see that only Gobi can successfully infer the known this risk factor causation. And furthermore, please look at that. Here, gray arrow is the result from when we use this 3 years time series data. The purple one is we only use the first 2 years data. Other method result is changing depending on the length of the data, but on Gobi, robustly infer the same regulation regardless of this data length. So yeah, that's all. So summary, Gobi can reconstruct the underlying network structure just from the time series data. This is a new type of causation detection method. And then density pin, they can infer from the time series data again, they can infer the time delay distribution, which can again tell the underlying network structure of the signaling pathway. Okay. So density pin was developed by Hyuk Hong and Dr. Zhou, and then Gobi is developed by the Sungmin Ha and Seo Park. And so a couple of years ago, we launched the Biomedical Mathematics Research Center in Korea, where George gave a talk a couple of years ago. This is a place where the various background people work together to solve the biomedical puzzle. We have some visiting scholar program for the faculty member, and we have a senior researcher and postdoc positions. And also, we have an undergraduate and graduate student internship program during the winter and summer vacation. So if you have an interest in, please contact me. And furthermore, we have an online colloquium series, like a crunch seminar. So I think one of the previous speaker was George Karniadakis. So this is open event. So if you have interest in how mathematics can be used to solve the biological puzzle, please welcome to join it. So and finally, this summer we have a International Society of Mathematical Biology meeting in Seoul. Already about 1,000 people registered. So if you have an interest in this quantitative biology, it'll be great if you want come to this exciting event in Seoul, in Korea. Yeah, thanks for your attention.
Thank you so much, Jae. Really, it was a really nice talk. Now, do you have any questions from the audience?
May I?
Yes, please.
Okay, thanks. This is Kostas
Mhm.
Hetos from the University of Naples. Thank you for your talk. A couple of questions, just of curiosity mainly,
Yeah,
from
sure.
both parts of your talk. So if you go to slide 13 on your signal transduction approximation.
13 here.
There, yeah. No, there. All right, good. This one, great.
Okay.
Okay, so
Yeah.
if you look close to the second time series on the right, on the bottom, it seems that after a transient, you have some oscillations, and oscillations are fundamental in mechanotransduction or signal transduction, which are associated also with oscillations in gene expression and multi-stability. But it seems that you fit it like an S curve, while I mean it seems that you fit it like you fit the transient. I mean, are you sure that it goes to a stationary state and these are not oscillations?
So number one, Kostas, thanks for the question. Number one, this is simulated time series data from this equation.
All right.
So indeed, this is not oscillation.
Still.
It's a fluctuation due to just noise. But as you
there seems that it's a frequency. It's more than noise. There is a frequency.
But this is just a Gillespie algorithm. We just served the chromograph equation using the Gillespie algorithm. So it's really fluctuation rather than oscillation. But indeed, in the certain experimental data, they are really oscillation.
Right.
That oscillation actually indicate that it's not following this simple birth death process.
Right.
It has a negative feedback loop. Then we need to a little bit modify this equation having the negative feedback loop. So here, this production term will also depending on the y of t. So in this case, we can still apply the density pin. We just need to change the this first production term to analyze oscillator time series data.
Right. Okay. Thanks. And the second question on your causality methodology and experiments, you saw some. Maybe I missed it. Some comparison with Granger causality, but Granger causality does not take into account spurious connections. I mean, did you consider, methods like full condition Granger causality and also nonlinear causality like mutual entropation, mutual information theory, and about also for the computational costs of this in multi-node networks?
So,
I mean, other than 4 or 5, I mean.
So actually, we tried actually a bunch of the Granger causality, but we got always this previous causation because please look at the data set. Here, 4 time series data actually has quite similar period, right? If you look at the left side graph, what is the major problem of the previous causality detection algorithm is they cannot distinguish the synchrony of the data and actual causation. So because they are based on the predictability, right?
There's also the partial Granger causality in the frequency domain that maybe can do it. Can identify phase differences. Okay, I mean just a notice about this. Yeah.
Okay.
All right. Thank you.
May I ask a question?
Great, please.
Sure.
Yes.
Hi. I'm Yu from Lehigh University. Yeah, thanks so much. Professor King for the.
You're from Ohio University. Nice to meet you.
Yeah. Nice to meet you. So my question is also about the causality part because I think you mentioned that in the previous part of your talk that if you use the Cindy, right? Cindy is not identifiable. So so basically, is your model the causality model? I mean, basically the dynamic causality model identifiable? Is that doable? Because I think
very good question.
Yeah.
You very good question. So the key is actually this map. So we need to know whole this domain is blue or not. That's the key to use the Gobi.
I see.
But if our data is too short, then what happens is only half of this triangle is filled, but half of the triangle may not be filled. In that case, we cannot tell.
I see. So basically, do you mean that
Yes,
the data basically with sufficient
which can
coverage, you will be able
yeah, of course, sufficient data. So the good thing of the Gobi is, unlike a Cindy, in the Cindy case, it's not easy to tell that it's identifiable or unidentifiable. But for the Gobi, if you plug in any type of data, they will try to fill this triangle. But if triangle is not filled enough, then Gobi will tell that we need more data.
Interesting.
We can clearly tell that we need more data or not just by looking at how much fraction of this triangle is filled.
I see. Yeah, that's very interesting. I'm also curious. So does that mean that it's identifiable such that you can know if this directed edge is pointing from X to Y or from Y to X?
Yes.
Coming from this kind of the regulation detection score.
Yes, correct.
I see. Yeah, that's super interesting. So basically, yeah, that's super interesting. Another question is also related with the, I think the density pins. So I'm just out of curiosity. So if you, can you go to like say your slide number 16?
16. Yes, actually.
Yeah, thank you.
Maybe this one, the structure, or
no. Can you get this 16?
16. Maybe this one is 16.
Yeah. Okay. Yeah, I think this one. So my question is, just out of curiosity, because you have the observation of y, right? So theoretically, you can calculate the derivative of y, basically the y prime and the y. You can know the integral of g tilde. So I'm just curious, if you do some deconvolution method for g tilde, does that work?
Oh, actually, we haven't thought about it, but that might be a good idea. Oh, I see. Just rather than y of t, use the y derivative t.
Yeah, right. Just substitute your observation.
I see.
So that becomes like the integral of is equal to something, right?
Yeah. I think there is, I think, pros and cons. One pros is if we use the y prime t, then I think the problem becomes much easier. But the better thing is usually experimental data is very sparse.
I see.
In that case, the derivative could be inaccurate information. But if we have a very dense data, I think using the y prime t information might be the really good idea.
Oh, I see.
Yeah. But thank you. But let me memorize and tell to Dr. Zhou. Maybe we can test this idea.
Oh, I see. Yeah. Thank you. I'm just out of curiosity because we also do learning, but we do that on the spatial data, not the temporal data.
I see.
Yeah. Thank you. Thanks a lot for the.
Thank you. Yeah.
So, Dr. Kim, I have a question. Can you hear me?
Sure. Yeah.
I have a question about the causality part. So, if you just go to that slide, I think you introduced some autonomous system. Yes, this one. Can you just go on back? Maybe
this one.
Maybe the next slide. Maybe sorry. Yeah, this one. Yeah, that one.
Yeah. Simple one. Yeah.
Just the previous one. Sorry.
Yeah.
And previous slide maybe. Yes, this one. So here, I could see an autonomous system here, but so when
if there is external input. Yeah,
if there is some sort of a time-dependent response, like how does it work? Because especially in case of like therapy situations, you have like like time dependent, some sort of like input.
So
can you recover the causality?
No. If we have some external variable, this Gobi doesn't work. But if we have that external variable time series data as well, then we can include it as a one more dimension. Then now it's a three-dimensional autonomous system, right? But of course, external variable is independent. Then then we can apply the Gobi.
Yeah, but I understand. But if it's something like, let's say, some sort of treatment, you know, like intervention on day 5 and 10 and 15, you just, you know, like give the drug and just, you know, like back up.
Then one way is if we know when the drug is given, then from the first day to the 4th day, we cut down that time series data. We know that it should be autonomous, right?
Oh, okay. That's a very good.
And then we apply the Gobi. And then from the 5th day to the 7th day, now the situation changed because there is an external variable, right? Then now we apply the Gobi to that dissect time series data. Then actually we can get the 2 different type of this regulation detection map, and both give information. So if we have a very long time series, actually, I skipped the explanation. For instance, this Hong Kong data, this is a 3-year data. So 3-year, there should be many changes, right? So how we apply the Gobi is we dissect this data as a 1 month or 1 week. We apply the Gobi for each window. And then for each window, we calculate regulation detection score, so that's why we usually get the distribution of the regulation detection score rather than just single value.
Oh, this is very interesting. Okay.
Yeah.
Okay. So I think this could be a very relevant situation, because that's usually almost all treatment protocols, like they're just implemented this way, actually.
Yes.
Thank you so much. Yeah, thank you.
Great. Do we have any other questions from the audience? It looks like we do not. In that case, I may ask a question.
Sure.
Can you go back to your densely pings the structure where you show the architecture?
Yes. Maybe.
That's faster.
Yeah, here we go.
Yeah, this one is good. I see that you have to train an encoder, a decoder, and you have another 2 fully connected networks.
Yes.
The first question would be: do you train them in parallel? Do you train the variational autoencoder at the same time as the other networks? Or
no.
You
so just serial way.
Oh, in a serial way. Okay, and the other question would be: how expensive is the computational time? Because you have to do backpropagation in 3 blocks, right?
Yeah, that's the good question. My answer is I don't know, because unfortunately simulation is done by the Dr. Zhou and Dr. Hong. I think they know the computation time, and but as far as I know, it's not that heavy. Kind of, for instance, to analyze that 16 the experimental data set, for instance, this cases, I think we, if I remember correctly, when I give this data set and when I came back next day, we got the results. So kind of, it's not the 1 week calculation, just the 1 day calculation is enough, I think.
Oh, okay. That's good.
Yeah.
Great. Yeah, so it's not that bad it's not that part. It's not bad. When I was worried because you have so many blocks, I thought that the computational time. Yeah, but it's nice because you manage to reconstruct all of this. Are there like some like numerical methods or traditional methods that can do something similar?
Oh no. So actually, to infer this time delay, actually previously, actually we working on this problem for a long time. Previously, what we did is we assumed that this G of S following some specific distribution like the gamma.
Okay.
Then we can use some Bayesian inference thing.
Oh, okay.
But so if we can write down what is this G of S, then like a G of S is gamma, alpha, and beta, then we can just need to estimate alpha and beta, right?
Yeah.
Then
Okay.
it's easy problem. But if we have no idea about the GS, then it's way more difficult problem. But this PIN was really great platform to do this.
Great. So you managed to do something that was not possible to do with other methods. So that's quite cool.
Yeah, it was really great. Yes.
Yeah, that's great. Okay. Do you have any more questions from the audience? It looks like we do not. So I think that I would like to thank you for your time. It was a really nice talk, and thank you so much for staying up late. Oh no, you said that you're in Hawaii, so you're not.
Yeah. Okay. So thanks so much for hosting.
Okay. Thank you so much, everyone. Until next weekend. Have a nice weekend.
Bye. Yeah. See you.
Bye.